Hello students, so welcome back to the channel. In this video, we are going to study about the transport of gases. See, two main gases, one is oxygen, another is carbon dioxide. So see carefully beta, transport of oxygen will be done by two methods. First of all, 97% of oxygen will combine with heme group. And you know heme group has iron. Remember, it has iron, so it combines with iron and heme group of hemoglobin molecule to form oxyhemoglobin. Clear? It will make oxyhemoglobin. So 97% is transported by the RBC. Only 3% is dissolved in plasma. See the line carefully. First of all, blood is the medium of transport of both gases, oxygen and carbon dioxide. 97% of oxygen is transported by RBC in the form of oxyhemoglobin, and 3% of oxygen is in the dissolved state through plasma. Clear, beta? This much is clear? Now see transport of carbon dioxide. 70% is carried in the form of bicarbonate. Okay, it is in the form of bicarbonate. 20 to 25% will combine with amine group. It will combine with amine group of globin molecule to form carba-amino hemoglobin. Clear, beta? And remaining 7% is dissolved in plasma. See the line carefully. 20 to 25% is transported by RBC. In which method? In the form of carba-amino hemoglobin. 70% is carried as bicarbonate ion. This only I told you. And 7% is in the dissolved state through plasma, dissolved in plasma. Okay, this is the number they are given it. Now, how the oxygen will be transported and how carbon dioxide will be transported, we'll study in detail now. Now, see, first of all, transport of oxygen. You know very well hemoglobin. Hemoglobin has two things. One is globin, another is heme. Globin is a protein part, heme is non-protein part, which is cofactor and it is prosthetically attached. It contains iron, that is why it is red color. This much is clear. And oxygen will bind with hemoglobin in a reversible manner. It is not irreversible, it is reversible. So oxygen will combine with hemoglobin to form oxyhemoglobin. So first of all, try to understand. Iron is present as Fe2+. When it combines with oxygen, valency of iron is not changing. To make oxyhemoglobin, valency of iron is not changing. That is why this process is known as oxygenation, not oxidation. Please understand it. It is not known as oxidation. It is known as oxygenation because we will not change the valency of the iron. Now, there are 4 heme groups are present in one hemoglobin. 4 heme in one hemoglobin molecule. And each heme has one iron. One iron in Fe2+. So it means one hemoglobin will have 4 iron. Logically clear? One iron will be able to transport one oxygen molecule. One iron means it will transport one oxygen molecule. So it means 4 iron means what? 4 oxygen molecule. Clear? It means one hemoglobin will be able to transport 4 oxygen molecule. Clear, beta? Because you know one hemoglobin has 4 iron and 4 iron means 4 oxygen molecule. So see carefully. Each hemoglobin can carry a maximum of 4 molecule of oxygen. Clear? Maximum it can carry how many oxygen molecule? 4. And binding of oxygen with hemoglobin, it depends mainly, primarily depends on partial pressure of oxygen. So binding of oxygen with hemoglobin is primarily, means mainly depend on partial pressure of oxygen, though other factor will also play a role. Other factor will also play a role. Which other factor? Like partial pressure of carbon dioxide will also play a role. Hydrogen ions will play a role. Okay, and temperature, they are also other factor which will interfere with this binding. This much you have understood. Now, let us study this diagram properly. See, when this is the partial pressure of oxygen. Please understand, this is partial pressure of oxygen, and this is how much hemoglobin is saturated. So they are giving the percentage of the hemoglobin saturation. Hemoglobin is, if you see in this graph carefully, it is 100% saturated. It is 100% saturated, and when the partial pressure is nearly, you can check this and you will match it coming here, means it is nearly 95, more than 95 partial pressure of oxygen. When the partial pressure of oxygen is more than 95 mmHg, then hemoglobin is 100% saturated. When hemoglobin is 50% saturated, if you see it is 50% saturation is here. See my green color, I'll use a blue color here. Please focus. Here the hemoglobin is 50% saturated, and the partial pressure at which hemoglobin will be 50% saturated, that partial pressure will come out nearly to be 28. Okay, because you know this line is 30. So this partial pressure is known as P50. Means at this partial pressure, hemoglobin is 50% saturated. So 28 partial pressure, 28 mmHg is required at the alveoli level. But at the tissue level, to maintain 50% saturation, not only oxygen, other factors also play a role. Carbon dioxide, hydrogen ions, temperatures. So when the carbon dioxide partial pressure increase, proton increase, temperature increase, in that case, the curve is shifting to the right side. So as the result, at the tissue level, okay, at the tissue level, P50, the partial pressure which is required to achieve 50% hemoglobin to be saturated is more than what it is required at the alveoli. So we say curve has shifted to the right, and this is known as Bohr's effect. Because right now we studied it is primarily affected by oxygen, but other factors also plays. Please focus. Binding of oxygen with hemoglobin is primarily related to partial pressure of oxygen, but other factors also play a role, like partial pressure of carbon dioxide, hydrogen ion concentration, and temperature. They are the other factors which can interfere with this binding. As a result, at tissue level, the curve will shift to the right. We say this is Bohr effect. It means now oxygen will be dissociated with hemoglobin. So see carefully. At tissue level, when the oxygen partial pressure is low, it means carbon dioxide partial pressure is high, proton concentration is high, and temperature is high. In such cases, the dissociation will go on. Dissociation of oxygen with hemoglobin will go on. But at the alveoli level, what will happen? At the alveoli level, partial pressure of oxygen is high, carbon dioxide is low, proton is low, and low temperature. All these factors will favor the formation of oxyhemoglobin. Clear, beta? This much I think so. What occurs at alveoli and what occurs at the tissue, I think so this is clear. You can just make a note this way. At the tissue level, obviously partial pressure of oxygen will be low. It means partial pressure of carbon dioxide will be high. Protons will be high. As a result, it means pH will be low and temperature is high. In these conditions, dissociation will occur. Favorable, it is favor for dissociation of oxygen from oxyhemoglobin. This much concept is clear to you. Now see, this type of curve is sigmoid curve we will obtain. sigmoid curve is obtained. I'm highlighting with the pink color. Please focus. Sigmoid curve is obtained when you are plotting percentage saturation of hemoglobin with oxygen against partial pressure of oxygen. Percentage saturation of hemoglobin with oxygen against this. So you are getting the S-shaped curve that is known as sigmoid curve. This is also known as oxygen dissociation curve. Please, it is also known as oxygen dissociation curve. Clear, beta? This much is clear, and you can, this curve is used to study what is the role of other factors like partial pressure of carbon dioxide, hydrogen ion concentration. Now, one more information, please understand it. In the circulation chapter, it is given that 100 ml of blood will carry nearly 12 to 16 gram of hemoglobin. 1 gram of hemoglobin will carry nearly 1.34 ml of oxygen. It means we will take round figure 15 gram. 15 gram of hemoglobin will carry 15 into 1.34 ml of oxygen. And I think so answer when you calculate it, I'll do in front of you, you can see 15 multiplied by 1.34, your answer is coming 20.1. It will come nearly 20.1, means we'll assume 20 ml of oxygen. The 15 gram of hemoglobin will carry 20 ml of oxygen, and 100 ml of blood has got 15 gram of hemoglobin. So can I say 100 ml of blood will carry, it will carry 20 ml of oxygen. Clear? And 100 ml of blood under normal condition will deliver only 5 ml. It will deliver only 5 ml of oxygen to the tissues. Please see this line carefully. Every 100 ml of oxygenated blood will deliver only 5 ml of oxygen to the tissue under normal physiological conditions. So I think so, oxygen dissociation curve and transport of oxygen, with all the calculation, this topic might be easy for you. Now let us study transport of carbon dioxide. Now for transport of carbon dioxide, try to understand. I'll just draw one diagram. Please understand it properly. This is your blood capillary, imagine, okay? This is the blood capillary and this is RBC. This is RBC. I'm drawing, this is at the tissue level. At the tissue level, carbon dioxide concentration is high, its partial pressure is high. So 7% of carbon dioxide will dissolve in plasma. Clear? It will dissolve in plasma. 23% of carbon dioxide, it will enter into the RBC and it will combine with amine group of globin molecule to form carba-aminohemoglobin. Here I will just try to focus. Oxygen was carried by the heme group, not by the globin part. Oxygen was carried by the iron of heme group and carbon dioxide is carried by the globin molecule of hemoglobin. Clear? Now remaining 70% is C. 70% of carbon dioxide will combine with water in the presence of an enzyme, carbonic anhydrase. Please focus, this is the enzyme which is the greatest quantities present inside RBC, but few quantities present in the plasma also. This is known as carbonic anhydrase. It will combine to form H2CO3, which breaks down to form H plus and SCO3 minus. Clear, beta? Now SCO3 minus will go out, and to maintain the electroneutrality, chlorine will come inside. And this is known as chloride shift. Chloride shift is also known as Hamburger phenomena. It is also known as Hamburger's phenomena. In the RBC, hemoglobin is combining with oxygen, which is combining with this one. Oxyhemoglobin is coming in this format. Now, when the partial pressure of oxygen is low, it means partial pressure of carbon dioxide is high. That time what happened? Dissociation will be there. Means oxygen will separate from hemoglobin and it will give into the tissue. Now, hemoglobin will combine. Please focus, hemoglobin will combine with proton to form hemoglobinic acid. And K will combine with the chlorine to form KCl. Is it clear? And bicarbonate ions, which has been diffused outside, this bicarbonate ions, they will do one thing, they will combine with the sodium ions. They will combine with the sodium ions to form sodium bicarbonate. Clear? To form sodium bicarbonate. So I think so 7% dissolved in plasma, 23% combines to form carboaminohemoglobin, and 70% is in the form of bicarbonate. Clear? This is at the tissue level. Now we will see the theory. See the NCERT. Carbon dioxide is carried by hemoglobin as carboaminohemoglobin, nearly 20 to 25%. And it is related to the partial pressure of carbon dioxide. Partial pressure of oxygen is the major factor which will affect this binding. Here the partial pressure of oxygen is decreasing. That is why oxygen is getting dissociated from the hemoglobin and hemoglobin is made available to combine with the carbon dioxide to form carboaminohemoglobin. Please understand the question. Partial pressure of oxygen is a major factor which will affect this binding to make carboaminohemoglobin. Clear, beta? Next, when partial pressure of carbon dioxide is high and this is low in tissue, more binding will be of the carbon dioxide. But in alveoli, what was there? If you remember, in alveoli, partial pressure of carbon dioxide was low and partial pressure of oxygen was high. So dissociation was going on. From carboaminohemoglobin was taking place. Okay. And carbon dioxide, which is bound to hemoglobin, will be released so that it can be diffused. So binding is primarily, major factor is oxygen. Major factor is oxygen. Now next, RBC contain high concentration of one enzyme, that is carbonic anhydrase. So RBC contain high concentration of carbonic anhydrase and minute quantity is present in plasma also. Please, very, very important question. Carbonic anhydrase is present in RBC as well as in plasma also, high in RBC. And this enzyme will do the reaction in the both direction. So carbon dioxide plus water will combine to form H2CO3, which dissociate to form bicarbonate ions and H+. So at the tissue level, what will happen? If you see at the tissue level, when the partial pressure of carbon dioxide is high, carbon dioxide will diffuse into the RBC and plasma and will make bicarbonate and H+. At the alveoli, it will be reverse. Entire equation what I taught you, we have to do reverse at the alveoli level. Means when the partial pressure of carbon dioxide is low, reaction will be in the opposite direction, leading to the formation of carbon dioxide and water. So it means carbon dioxide is converted as bicarbonate ions at the tissue level. Carbon dioxide is trapped as bicarbonate at tissue level and is transported in the alveoli back into the carbon dioxide and water. Clear, beta? Now, 100 ml of deoxygenated blood will deliver only 4 ml of carbon dioxide to the alveoli. So this is the transport of carbon dioxide. So I hope so each and every line of NCERT I have focused it. This video will be helpful for you. Please learn NCERT properly. Thanks for watching.